There we are. Okay, so let's get started. Good morning, everyone. So first of all, I would like to thank the organizers for inviting me and giving this talk on perinatal stem cells, the gold mine. I've divided my talk into two parts. So first of all, I would like to give you a small overview what is going on in terms of the theory of placenta stem cells. And in the second part, I'm going to show you some clinical examples. And if you are still alive by then, we can answer the questions. So, the placenta. Actually, it was supposed to be a waste product. It has been thrown away. And when you look at that, you can see this is the research from PubMed, all the papers that have been published in the last, let's say, 5 or 10 years on placenta. And you can see there's a huge increase of papers being published on that topic, and the numbers are still going up exponentially. So what is the most interesting part when looking at that waste product? So first of all, what we know so far is that the cells we have in the placenta are young, they are healthy, they had no contact to any environment, no toxins, nothing. And these cells do not have any immunogenicity. That is the very important part here. They can be used for the secretion of cytokines. I'm going to show you a very striking example in a few moments. And these placenta cells do play a significant role, not only in tissue repair, myocardial infarction, and first of all, tissue repair, regeneration, and cancer therapies. I'm a hematologist, oncologist by training, so we are looking for cancer therapies using placenta stem cells. And they are also able to correct genetic disorders. So when we are looking, and you know that better than I do, when we look at placenta, placenta is a very complicated organ in terms of cell population. So we have several cells here. We have the trophoblastic cells, we have stromal cells, chorionic cells, amniotic cells, MSCs, mesenchymal stem cells, and we have exosomes. And you take a placenta and you shake it, and you have a tissue, a petri dish. You can select and you can isolate plenty of exosomes. And these are very, very important for several therapeutic indications. So the most important thing when talking about placenta cells is the fact that they do not, all types of placenta cells and even the normal stem cells derived from cord blood, they do not express HLA-2 markers, DR, DQ, DP, and they don't express any co-stimulatory markers such as CD80, CD88, CD40. So we don't see any graft versus host reaction. Nothing at all. They are simply immune evasive. And as I mentioned earlier, you can use them, especially the mesenchymal stem cells, as the most advanced cell population. You can use them for tissue repair or tissue regeneration. And you can use them as novel anti-cancer agents. So what are the procedures you normally do? You can see on the left-hand side, you see the placenta coming into the lab. Then we look, as you do at birth as well, to make sure that the placenta is complete. Then we are looking for the specific types of tissue, which is shown on the upper right. And then we are going to cut it into pieces, and then we are digesting that tissue, and we can isolate certain cell populations, which are shown on the bottom right. So these cells are then, these cells can then be used for any therapeutic application. So that is a nice cartoon stemming from an Italian group, and here it is clearly indicated what can be done. You see a placenta stem cell on the left, and these small bubbles here are exosomes derived from the placenta tissue, and you can see you can use them for tissue regeneration, you can use them as anti-tumor therapy, and you can use them for the correction of any genetic disorders. So during this talk, I'm going to focus on the tissue repair and on the anti-cancer situation as an anti-cancer drug. The good thing here is taken into account that these are young, healthy cells, and I've shown it on the left-hand side, the amniotic cells. They can differentiate into pancreas cells, bone, liver, CNS, cornea, and the same applies with the mesenchymal stem cells. You can drive them into myocardial cells. You can drive them, again, liver, bone, and all the tissues you probably want to have. And this is from one of our publications for the MSCs, and you can see this is, don't need to look at that in detail, but you can see tissue replacement, and in the green box you can see what can be done here, and I'm going to show you a striking example in a minute. And of course you can see tissue repair. Let's say someone who swims in alcohol and has a liver cirrhosis. You can correct liver cirrhosis, at least in the animal model, using these stem cells. So that is shown here. This is one of my PhD students, and you can see these are chorionic cells taken from a normal placenta, an organ that is thrown away. But now we have isolated stem cells, and you put them in a certain environment, growth factors, steroids, depending on what you want to have. And then you can see you can differentiate these cells into adipocytes on the left-hand side. You can differentiate them into chondrocytes, chondroblasts, and you can differentiate them into osteoblasts, for instance. This is depending on the environment the cells are living. So you take the cells, put them in a certain growth solution, depending on what you want to have, and then you end up with a certain cell subpopulation. So the most interesting part of human placenta are the extracellular vesicles, the exosomes. We have 3 of them in terms of molecular biology. We have the apoptotic ones, these are the bigger ones. We have the ectosomes, medium size, and we have the exosomes. The exosomes are, again, not immune, they are immune evasive. They don't cause any reaction, as they do not express HLA, class 2, CD40, CD80, CD86, and they can easily stored. Just shake the placenta, strictly speaking, shake them when they're falling out. You can obtain them by that. You can process them and store them at -80. No problem. Plus 5% DMSO. And they can be used to restore damaged tissue, and they have, as I mentioned earlier, a huge benefit, at least in vivo, for the treatment of cancer and genetic disorders. So why are these exosomes so interesting? They're interesting because they contain so-called micro-DNA. If you take a mirror this morning in a bathroom, and you look into the mirror, you see your face, how you look. 50% of the way you are looking, 50%, is due to microRNA. So microRNA in your body is responsible for the way you look, independent of what you're doing. But the way you are looking is in 50% based on microRNA. And these are actually non-coding RNA molecules, very small molecules, and they are responsible to regulate gene expression. So they can shut off certain genes, they can shut on certain genes, and they can, by doing so, controlling gene expression patterns. And this is very important because you have roughly 2,300 different microRNAs. And the good thing is now, some of these microRNAs can be used for correction of genetic disorders. Some of them can be used for cancer therapy. And what you can do, you can load the exosomes in vivo with these microsomes and microRNAs. You can put, as an experimental system, you can put microRNA into exosomes, and you can use these exosomes with specific microRNAs for a treatment application. And the microRNA 124, for instance, is one that we normally use as cancer treatment. So here it's an example. You can see here on the bottom left, this blue bubble is a stem cell taken from the placenta, and you can see these stem cells secrete these exosomes, and the electronic microscope picture is shown there. And then you can load them with specific, very specific microRNAs, and then you can use them to repair injured cells, using them for tissue repair and so on. Okay, let's give you a striking example. This is NASH, non-alcoholic liver cirrhosis. And when you see, you can use an animal model, you can get a mouse to develop liver cirrhosis. It's just a question of diet. And you can see on the bottom left, you can see the typical histologic picture of a liver cirrhosis. And then we have treated these animals with exosomes loaded with a specific microRNA. And what you can see here, with time, and you see on the bottom right, after 3 weeks, you can see the liver cirrhosis in this animal model is resolved. This is a normal mouse liver, no liver cirrhosis at all. Okay, and when using that, and I mentioned it before, you can go for the microRNA 124. You can use that for cancer treatment. And just look at the bottom left. These are 3 different tumors in vivo, a nude mice model, and hepatocellular carcinoma, malignant lymphoma in red, and the glioblastomas. So these tumors have been treated. That is important. These tumors have been treated with exosomes taken from a normal placenta, loaded with specific microRNAs. And what you can see here on the bottom line, the upper 3 are the controls, untreated mice, and you see the bottom one, the tumors have vanished. And ladies and gentlemen, these tumors never come back. So, this is the most important co-worker in our lab. It's a mouse. So this is taken from a clinical trial in Jerusalem. We are recruiting patients for that trial. This is an experimental model. And the mice have been treated with a single shot of irradiation, 7.7 gray. It is 7.7 because it has been done in the US. They like it a little bit complicated. So 7.7 as a single shot. You know, from the clinic, you normally give a fractionated irradiation, 1.8 up to 2 gray, and every day. Now these animals received 7.7 as a single shot. This is a lot. The helicopter pilots at Chernobyl, the first ones who put the concrete on the reactor, received 12, and they're all dead. And these animals received 7.7. Then, what we did here, or what the colleagues did, on day 1 and day 5, they received stem cells derived from a human placenta. So we transferred human cells on these not immune-compromised mice. And you can see these cells are here on the left. This is the electronic microscope picture. So we put it on there. Yeah. I'm not crazy. I'm telling you, we're taking human placenta cells and transplanted them into mice. We could do it the other way around, but this is not ethical. But anyway, so, and this is the result. So, what you normally see when you're going to irradiate an animal with 7.7 gray, the bone marrow is wiped out. There is nothing left. And here you can see, as proof of concept, only 23% of the animals survived. If they received the human placenta stem cells, you can see 81% survived. Mice with a human placenta cell preparation. And this is, or was, the basic data for a phase 1 trial published 2 years ago at the American Society of Hematology. And they had 19 patients, And these patients had actually had no hematologic reconstitution after myoablative chemotherapy after 3 months. Normally it's day 100. We have to stick to day 100. And you can see hemoglobin below 8, neutrophils below 1,000, thrombocytes below 50,000. We call that a graft failure. Roughly 25 to 27 patients following bone marrow transplantation have to cope with graft failures. So these patients were then treated, 3 cohorts, with human placenta cells, taken from any placenta at all, because they are immune evasive. There's no graft versus host reaction. You can take any placenta at all. And you see low dose, intermediate dose, and high dose. Again, and these cells were given intramuscular. Not even in the vein, you can put them in the muscle. And what we can see here, in this phase 1 trial, there was a significant improvement of all 3 cell parameters, and the treatment was very well tolerated. No graft versus host reaction, nothing, just a minor erythema around the injection site. And the phase 2 study, where we are going to enroll patients, is ongoing. So let me come back. This is the situation you might know from your lab experience. So I'm now coming back and now going to the clinical part and to demonstrate some clinical examples. So that is where we use, in that case, cord blood stem cells. In this case, for Crohn's disease, there was a large study published some years ago with 108 patients. I'm not going into the detail. The patients who actually were randomized Crohn's disease between placental stem cells and steroids. Standard procedure. So what you can see here, on the left side you see CDAE, that's Crohn's Disease Activity Index. So what we see here with the steroid ones, the dotted line, not that much improvement, and you see the solid line, there is a major improvement of Crohn's Disease Activity Index. That looks theoretical, but I have the pictures here as well. And you see on the left-hand side, A and C, you can see the endoscopy. You see there is some sort of inflation that is not the normal mucosa, what you would expect. And after giving placenta stem cells, you see on the right, this is a perfectly normal picture situation when you do an endoscopy. All done with placenta cells, in that case with cord blood stem cells. So there is another very interesting study, and there are tons of studies. I'm just picking some relevant ones. This is the chronic kidney failure. This was a randomized placebo-controlled trial with 40 patients, and they were treated, these patients were treated with placenta-derived exosomes I mentioned before, 100 microgram per kg, day 1 and day 8. And what you see after 12 months, no adverse events, decrease of creatinine levels by 50%, and increase of the GFR twofold. And these are the data you see on the left-hand side. You put the catheter into the renal arteria and you inject these exosomes in both kidneys. And you can see on the upper right, you can see the creatinine is going down, and down on the red bars, you can see that the GFR is going up. So, and that is the histology. That is very interesting. We have done that in mice as well. And you can see on the left-hand side, you see the control. And then what you see here, you have, in the second one, you see the picture untreated, acute renal dysfunction. And then you add the exosomes, and you can see on the right-hand side as a complete restorement of the histology in these patients. The bottleneck. Well, there are two sides of each coin. The bottleneck is we don't have enough cells. We have to be very careful what we are going to do with the cells, and we have to be very, very careful how many cells we get out of a given placenta. So, and there's recently, it has been approved a new compound, and you need a language course to pronounce that. It is omidubicil. Omidubicil has received the FDA approval in April this year, and we expect the approval from the EMA with a positive champ votum at the end of the year. This is a new method that allows us for a ex vivo expansion of cord blood cells as it stands right now, but the same method, and this has been confirmed by the manufacturer yesterday, that can be used for any placenta cells as well. This is a cell biology technique. I'm not going to bore you with what happens here. And this compound has been approved currently for the allogenetic application. It means you can take a given placenta or cord blood from everyone, and you can send it to the manufacturer, and they are going to expand these cells up to 1.10 to the 8, and then you can use them as a source for bone marrow reconstitution. Currently, using cord blood, we can only treat children up to 10 kilograms, because the bigger ones, you or me, there are not enough cells. So to have that bottleneck solved, we are now able to expand these cells. You just send the cord blood taken from the cord, you send it to the manufacturer, and they are going to prepare and to expand it, and you end up with 1 times 10 to the 8 cells. So that is something that is brand new, and that actually helps us to open the bottleneck. So that is the clinical data of the registrational trial. It took the company more than 12 years to get that product approved. And you can see on the left graph, the blue one, shows you the hematologic reconstitution using omidodubicel expanded cells, and the right one are normal cord blood cells. And you can see there's a significant difference. And the most important thing is, even after less than 7 days, we have an engraftment. This is a very important thing, because these patients do not have any immune system at all. Every day we have the earlier the hematologic engraftment, the better for the patient. So that is a really breakthrough, and the FDA mentioned that as well. So coming to the end, stem cells, clinical trials. These are all the cells we are currently using, and you see the status on the right-hand side. There's no need to take any photographs. You can download it and whatever. You see 2 of them are preclinical, the trophoblastic cells. They are now coming more and more in the focus of development. They represent, by the way, a model for cancer metastasis, and they can be used for growth retardation in mice, for instance, and so on. The chorionic cells are the most potent cell type, by the way, for tissue repair and regeneration. They are still preclinical. And then the stromal cell, the mesenchymal stem cells, amniotic cells, and exosomes are all in phase 1 or even phase 3 clinical trials. And some of them have been already approved. You can use them. And I would like to draw your attention to the expanded MSC from cord blood. These can be used, for instance, for cartilage degeneration. And it does work. Look at me. In this knee, these cells are in. The surgeon wanted to have me shifting me in the operation theater, and I said, no way. No way. We are doing it like this. I was a terrorist on the tennis court, and I tried very hard. I played a very hard return or whatever, and therefore I had the problem with the knee, and we put these cells into my right knee, and since then I'm not allowed to play tennis, but I'm happy and can walk around and go to the bar and drink beer. So, take-home message for you, those who survived the talk. There are no, and that is the most interesting part, there are no HLA expression in the placenta stem cells, exosomes, and cord blood cells. And they can be applied without HLA matching, allogenetic. You can use them without any immune suppression. And that will allow, that is the near future, you will have here a off-the-shelf drug in the near future, and omibubicel is one of the first examples. You can think of putative indications, tissue repair, you can think of correction of genetic disorders, and of course of cancer therapy. And overall, no graft versus host was seen in any of the clinical trials so far. And remember, you can transplant them without any precautions from human to mice, and most likely vice versa. But so, when you look at the famous painter, Lucas Cranach, and they have done a famous painting. If you own it, you don't need to work any longer. So the fountain of youth, and you can see on the left-hand side, all these guys are going into that swimming pool, and there is some sort of rejuvenation, and they get out, and they are young, no disease at all. This is a vision. We are not there, but we are at least in the middle of the pool. So. And when you look for Nelson Mandela, and that is my personal opinion, and I'm doing that for 10 years, placenta, the secret of success in life is to be ready when the opportunity comes. So that's why I'm asking you and I'm pledging, do not throw this organ away. It is a gold mine for the patients right now and most likely in the future. Thank you very much for your attention. Questions? There is one. You can go to the microphone, it's probably easier. Therefore, they are there.
Is there any study concerning infertility, ovarian premature failure, or aging-correlated infertility with the stem cell or a placenta product?
There, at least in vivo, for the trophoblastic cell population, we know that growth retardation can be corrected, and we can correct preeclampsia. For other diseases, there are no data at all and no clinical trials ongoing. Other questions? How about you? No?
How are the laws regarding the donation of the placenta tissue? Can everyone just donate it for we go into treatment?
What are you normally doing? You throw them away.
Yeah.
Oh, you can. There are three opportunities. You throw them away. The purists actually eat them. Mm-hmm. Yeah, and you put them under an apple tree. That is the three options you have. So what you can do is there are this man there in the blue jacket. He is the chief executive officer. You can store them for ages. And that costs how much? For 20 years, 30 years, 25 years. So the good thing is you have nuggets. Yeah? Nuggets, gold nuggets in the freezer. And you don't know what happened in 5 years or 10 years. The knowledge in medicine increases exponentially. And then you have that cell population in the freezer, and you can use that for whatever you want to know and what you want to do. And you see, we are now able to expand cells using that homeobicil. So that is something that is of great interest. And I'll give you an example. There is a healthcare insurer in Germany, Allianz. It's here in Geneva as well. They said, okay, we are donating health insurance for patients by storing placenta. They're taking over the costs. The project is in preparation. We are taking over the costs, so the parents have the placenta stored, and they can be used for whatever. That is the future, rather than throwing them away or eating them. Any other questions?
Just one thing to that, the situation in Switzerland is as such that at the moment there is no possibility to really, let's say, donate it and store it in Switzerland, except for you can donate it for research and development in some university, the University Hospital in Zurich, for example, or you have the possibility with our company to actually store it, but then it's a private storage. We're not a public bank, then it's only, say, for the family or for the donor itself.
Gunther, can you allude a little bit about the procedure what parents have to do?
Sure. The way that it works is that if families decide to do so and store the placenta, then basically they first of all they make a contract with us, and then they get a box which is about that size, and it's a collection box where all of the necessary tools are in there. They take that box to the hospital, and then the gynecologist and the midwives are trained. They know what they need to do, and then they basically take the placenta, also the cord or the cord tissue and the cord blood, if that's also asked for, and then basically that's being transported into our lab. It's then treated and prepared, and then it's cryogenically stored with a minus 190 for 25 years. And then basically, if any therapies or could be used for therapies, it will then be released, and it could be sent worldwide. So there is also a small dry shipper, it's called, because the cooling chain can never be broken, because if the cooling chain is broken, then of course the samples are gone, it's destroyed. So if a transplantation is needed, then basically it can be transported to any transplantation center in the world with such a dry shipper in liquid nitrogen. That's how the procedure works.
Any other questions? So take-home message, don't throw it away, and don't eat it, and don't put it under the apple tree. Just store it. It's a gold mine. Thank you very much.